Good morning, everyone. And then we have also completed amino acids. All of you are present. How many of you are not present? In amino acid class? Only one, two, three. Okay. And then the plasma proteins has also been completed. I think in this week. Plasma proteins. How many of you have not done that class? So all of you did that class. Do you know what are plasma proteins? What are plasma proteins? Exactly. Those proteins which are present in plasma. Can you name some of them? Good. So today we will be starting with the proteins and the peptides. So first, you have already studied about the amino acids. So can any one of you tell me the names of some amino acids? Lysine, cysteine, yes. So how many amino acids are mainly present? What are the number of important amino acids? 20. What are their names? And? Oh. So when we are considering proteins and peptides, so which one has the more number of amino acids, proteins or peptides? Proteins. So we generally consider that all those peptides which contain more than 50 amino acids will be called proteins. Or sometimes we also consider those amino acids which have molecular weights more than 10,000. That can be called protein. So today we will also learn about the different structural organizations of proteins. So there are 4 types of structural organization. Primary structure, secondary, tertiary and quaternary. So when one amino acid is connected to another amino acid, it is peptide bond. So when 2 amino acids are connected, so you will call it a dipeptide or monopeptide, because there is only one peptide bond connecting 2 amino acids. Dipeptide, but how many peptide bonds are there? One. So it doesn't matter how many peptide bonds are there. It depends on the number of amino acids. So it is tripeptide. How many peptide bonds will be there? 2. So first, when 2 amino acids are present, it is a dipeptide. And this reaction between 2 amino acids being connected by a peptide bond is called condensation reaction. Here, one molecule of water will be eliminated when the peptide bond is being formed. So similarly, we have tripeptide. There will be 3 amino acids. Tetrapeptide. There will be 4 amino acids. Penta. There will be 5 amino acids. So in the last class, I think you have also learned about the functions of protein. So can any one of you tell me some functions of protein? Transport, then?
The immunization and the antibody proteins.
Antibody. The antibodies are also protein. Neurotransmitter. Collagen. Then, what is the function of collagen? It mainly helps in the formation of structure and extracellular matrix. Any other function? Enzymes. So enzymes are protein in nature. Then what is the most important thing? That is the muscle. The muscle in our body is made up of protein. And when we contract our muscles, it is the contractile proteins. They are actin, myosin. So you have to know about the functions of protein. And then this every protein, they are being formed by amino acids. And the amino acids can be classified in several ways. So there are positive amino acids and negative amino acids. Means positively charged and negatively charged. Can anyone of you tell me some negatively charged amino acids?
Aspartic acid.
And glutamic acid. And what are the positively charged amino acids?
Lysine,
Arginine, lysine, and histidine. So this charge characteristics helps to identify what kind of amino acid it is. And when we try to find out the structure of a protein, we have to analyze how many amino acids are there and what are the characteristics of these amino acids, so that we can isolate those amino acids from the proteins. So first, 2 amino acids is connected by a peptide bond. So then there are oligopeptides. Generally a few amino acids, about 9 to 10 up to. And after 10 amino acids, if a polypeptide chain contains, it is called polypeptide. Below 10 amino acids, it is oligopeptide. And I have already mentioned when the molecular weight is below 10,000, these are polypeptides. And when above this number, these are proteins. Or we can also say between 10 to 50 amino acids, they are called polypeptides. And more than 50 amino acids, these are proteins. So here, so what is the normal structure of an amino acid? What is the backbone formed by? In any amino acid?
So one amino group, one carboxylic group, and one hydrogen bond. 3 bonds of carbon.
One amino group,
One
one carboxylic group, and
one hydrogen and one alkyl group.
there is another carbon. So what is that carbon called? Which is connected to alpha carbon. So the carbon just next to the carboxyl group is alpha carbon. So we say that NCC backbone. So if you see here, this one is the N, this is C, this is the alpha carbon, this is also C. Then when these 2 amino acids are connected, so here we can see NCC, then NCC. So this forms the backbone. And what is this R1?
Alkyl group.
This is the side chain of an amino acid. So this gets changed in every amino acid. So this R1 is one side chain, and when this side chain is changed, this is R2. This is another amino acid. So here also we can see that which in this polypeptide or in this dipeptide, this is the peptide bond here. So which are the structures or the molecules which can contain charge?
Amino, nitrogen and carboxylic carbon.
They can contain charge. And some other groups that can contain charge. So what are the other groups here? They are the side chains. Yes, sometimes, not always. So these parts, they are also containing charge sometimes, but not always. So here we can see, this is the C alpha carbon. This is the C alpha carbon. So here one side chain is attached R. Here also another side chain R is attached. So they can be same amino acids or can be different depending on the structure of R group. So this, what is this bond? This is the peptide bond between this amino acid and this amino acid. Here also, this is the C alpha carbon. So here this one, this is the peptide bond. So here we can see that the arrow is shown here. So that this bond, this, what is this bond? N C alpha. So this bond can rotate, but not this peptide bond. Here also we can see this bond, C alpha C. This bond can also rotate. So due to this rotating power, they can form different structural conformations. So this is pi angle and this is psi angle. But in peptide bond, they are partial double bonding nature. They cannot rotate. So now we come to the structural organizations. So as I have mentioned, there are 4 levels of structural organization: primary, secondary, tertiary, and quaternary. So first, primary structure. They are the amino acids connected by peptide bond. And sometimes this primary structure can also contain another kind of bond, that is disulfide. So if there is any change in the sequence or the number of amino acids in a polypeptide chain, the characteristics of the protein can change, and that can lead to different diseases. So here in primary structure, 2 things are important. One is sequence and the number of amino acids. They are connected by peptide bond. And this primary structure can also sometimes contain what kind of bond? I just said. Well, disulfide bonds. So now we come to the secondary structure. This is just twisting of the polypeptide chains, and there are several other kinds of bonds which helps in the formation of the secondary structure. That can be electrostatic forces, van der Waals forces, hydrophobic, and hydrogen bonds. So after the secondary structure, we come to the tertiary structure. In tertiary structure, this forms the functional structure of the polypeptide. And when several polypeptide chains are present in a protein, then it becomes quaternary structure. So what are the main kinds of elements present in a protein? That can be carbon, hydrogen, oxygen, nitrogen, and sulfur. Can any one of you tell me any amino acid which contains sulfur? Anyone? Cysteine, methionine, and cysteine. Cysteine, cysteine, and methionine. So now we come to the primary structure. The sequence is very important. And when we know the sequence of a normal protein structure, then we can only identify the diseased polypeptide or the diseased protein. So we have to know that which one is normal and which one is diseased, and which amino acid has been changed in the sequence or in the order. So what is the normal structure of RBC? The red blood cells. So it is circular and flattened at the middle. And there is a disease called sickle cell anemia. So any one of you know what is the sickle cell anemia?
Sir, ওই অ্যামাইনো অ্যাসিড সিকোয়েন্সের 6 পজিশনে GLU এর বদলে গ্যালাইন হয়ে যায়, চেঞ্জ হয়ে যায়।
In hemoglobin there are alpha chains, alpha polypeptide chain and beta polypeptide chain. In beta polypeptide chain, when there is mutation, then point mutation, then the protein gets changed. So then, ঠিক আছে, তবে ক্লাসে হাততালি দেবে না? না, ভালো বলেছি তো। So when the structure of this protein gets changed, and when this hemoglobin is in a hypoxic condition. So what is hypoxia? When there is less oxygen. Anoxia is there is no oxygen. Hypoxia is less oxygen. So then this hemoglobin gets clumped and they form a fiber-like structure. And this structure changes the structure of a RBC cell. So it becomes the shape of a sickle. And when this shape gets changed, these RBC cells cannot pass through the small blood vessels. So they get trapped within the small blood vessels and get broken down. And that causes hemolysis. So this disease is called anemia. So when the red cells are being broken down inside the body, so the number of red cells will come down. So what is the definition of anemia? Decreased capacity of the blood to transport oxygen. That is the normal physiological definition of anemia. So when there is less RBC, there will be less hemoglobin, and there will be less transport of oxygen. So this is sickle cell anemia. So here we can see there is a mutation. So the protein is getting changed due to the changes one amino acid. So here we can see it is the beta chain of hemoglobin. So there is in deoxyhemoglobin, that is hypoxic condition, the shape is changed and they are forming a fiber-like structure. And this causes the change in the shape of the RBC. So this is the structure of insulin. So what is insulin? Hormone. It is secreted by which gland? Pancreas. Endocrine or exocrine? Endocrine part of the plant. So here we can see there are two chains. One is A chain and B chain. So this is A polypeptide chain. So here there is no C chain. In this picture there is only A chain and B chain. So in A chain, how many amino acids are there? 21. And in B chain, 30 amino acids. So in this structure we can see there are also disulfide linkages. So here is one disulfide linkage, here is another, and here is another. So in this structure, there are two kinds of disulfide bonds. Can anyone tell me what are the two types? Interchain and intrachain. So this is the intrachain disulfide linkage, and this is the interchain. These two. So the peptide bonds. So peptide bond, these two amino acids, valine and alanine, they are connected by peptide bond, and they are forming valine alanine. So here we can see this amino acid, quaternary. Class quaternary. So here we can see these two amino acids is connected, and the end of this amino acid is getting changed to YL, valine alanine. And this peptide bond, this cannot be broken down by just heating or high concentration of urea. So later we will study about denaturation of protein. So when we heat a protein or a functional protein or an enzyme, so the structure of the protein changes because the bonds what we have studied are broken down. So which bonds can be broken down by heating? Hydrogen bond, van der Waals forces, electrostatic bond. But this peptide bond, they cannot be broken by just heating. To break these peptide bonds, we need strong acids, and we have to heat it for a long time to break these peptide bonds. So here also the same thing, these two amino acids are forming the peptide bond, and the name gets changed. There is also another thing. We have to know what are trans and what are cis peptide bonds. So when two amino acids are getting connected, I have mentioned only one thing changes if the amino acids are different, and one structure remains the same in all the amino acids. So which part changes when the amino acid changes? R part. So here we can see this one is one amino acid here, and this one is another amino acid. They are connected by this peptide bond. So one R is on downward side and one R is upwards. So when the two R groups are on different sides or on the different planes of the peptide bond, then they are trans. And when these R groups are on the same side, they are called cis. So in a normal structure of a polypeptide, which is favorable, cis or trans? Trans. Any reason? Steric. Because if two groups are coming too close, they are getting crowded. So what the normal tendency is to prevent this crowding, these R groups are in opposite directions. So trans is more common. So here also the same thing, when we are writing any polypeptide chain or when we are writing the structure of any amino acid, we always write the amino group on the left side and the carboxyl group on the right side. This is the general rule. Sometimes in books it can be reversed, but that is not the general rule. The general rule is we always write the amino group on the left side and the carboxyl group on the right side. This is also true for a polypeptide chain. So here this is a polypeptide chain. There are 4 amino acids, R1, R2, R3 and R4. We are writing the amino group on the left and carboxyl group on the right. And when the 2 amino acids are being connected with the peptide bond, the end of the first amino acids, whatever the end is, in, and, ik or it is changed to il. And when the amino acids are connected, they are not called as amino acids, they are called the residues. The residues in a polypeptide chain. So the characteristics, they are partial double bonding character. And other than the peptide bond, the other bonds can rotate. So here we can see, this is the peptide bond. So normally the distance is about 1.33 angstrom. But when a normal single bond is there, the length is higher. And when a normal double bond is there, the length is lower. So this peptide bond is in between this single bond and double bond. That's why it is called partial double bond character. So these peptide bonds are also uncharged and polar in nature. And what are the charge groups that can be present? That is the N-terminal amino group or carboxyl group. So this peptide bond has uncharged and polar character. So what are the charge groups? The terminal amino group or the terminal carboxyl group. And the other charge groups can be on the side chains. So this peptide bond can also sometimes show resonance. So what is resonance? This pair of electrons can change its position from here to here. So that's why in these 2 pictures, they are interconvergent. So in this first one, the double bond is here. And it can change the side of the double bond to here. So this characteristic is called resonance. So in this picture, this is a polypeptide. So what kind of polypeptide it is? Di, tri, tetra, because there are 4 amino acids. These are the side chains of these amino acids. And these are the peptide bonds. And this peptide bond can have some characteristics called dipole moment. Just remember that peptide bond can have a characteristics called dipole moment. Now we come to the primary structure. How to determine the primary structure. So what are the 2 important things which are characteristics of primary structure? One is the number of amino acids and sequence of the amino acids. So first we are determining the number of amino acids. So what we have to do, we will take a polypeptide and then we have to heat it with a strong acid so that the peptide bond breaks. So after the polypeptide chain is broken down into all its component amino acids. Then we use some technique called chromatography. So there are different kinds of chromatography. Here we are using ion exchange chromatography. So in ion exchange chromatography, there can be cation exchange chromatography and anion exchange chromatography. So there are other kinds of chromatography also, like paper chromatography, HPLC, high pressure liquid chromatography. But here we are using cation exchange chromatography. So what will be its function? So the column will be charged either positively or negatively. And we also know that depending on the characteristics of the amino acids, a protein can also be positively charged or negatively charged. So when we are breaking the peptide bonds, we are separating the amino acids. And then when the positively charged amino acid will pass through a column, it can be taken in depending on the charge of the column. So first thing is hydrolyzing the bonds, the peptide bonds. And then we are using this chromatography technique. Then after the amino acids are getting stuck in the column, the chromatography column, they are being eluted. Elution is the word, like we have to wash our desired amino acid out of the column, and we have to take it out, and then we will measure the concentration of that specific amino acid by passing light through the liquid where the amino acids are present. So first tell me one thing, when we are passing some kind of wavelength of light through a less dense solution and a more dense solution, which solution will transmit or pass more light? The less dense, because the more dense solution will absorb more light. So this technique is called spectrophotometry. So here first we are collecting the separated amino acids, and then these amino acids are reacted with a chemical called hydrogen, so that color is formed. And then with this spectrophotometry, we are characteristically finding the concentration of the amino acids. So in this picture we can see, first we have already separated the amino acids by lysing the peptide bond. Now we are injecting the amino acid solution. So these different amino acids are getting stuck at different levels. Then we are using a buffer or the eluting solution. So when we are sending this solution, one by one, these different amino acids are washing away from this column and passing through this coil. When the amino acids are passing through this coil, we are sending the hydrogen chemical, so that they are giving a reaction. And when this solution is coming out, we are passing this solution through spectrophotometer. So this is the photometer, and this is the source of light, so that we can estimate the concentration of the different amino acids, like how many amino acids are present. So here, what we are finding, the number of amino acids in a protein. But in the primary structure, there is another thing important, that is the sequence. So after finding the number of amino acids, we have to find out which is the order in which the amino acids are connected by the peptide bond. So here we will be finding about the sequence. So we will start from the end terminal. So there is some reagent called Edmond's reagent or phenyl isothiocyanate. So this chemical will react with the N-terminal amino acids in a polypeptide chain. And this reaction forms instability to the next peptide bond. And when this instability is inserted in the peptide bond, we can break only that peptide bond having the instability. So here we can see this is a polypeptide chain. So this is the N-terminal and this is the C-terminal. So here in the N-terminal, this chemical is reacting, and where will be the instability inserted? This is just here, because this is one amino acid, this is another amino acid. So here we can see, after this instability is inserted, this amino acid along with this chemical phenyl isothiocyanate is getting separated, and the remaining peptide chain is here. So one by one we can insert instability in the peptide bond and then break it, so that when one by one amino acids are coming out, we can say this one is the first one, this one is the second one, this one is the third one. So here also you can see this is a polypeptide chain containing 5 amino acids. So here the instability is inserted. So first this amino acid is coming out. So one is labeling and one is released. And once this amino acid is released, we have to identify what kind of amino acid it is, whether it is alanine, whether it is valine, whether it is glutamic acid or something else. So for that we are using another chromatography technique, that is HPLC. So this way we can identify the sequence. So first tell me another thing, how any protein inside our body is synthesized.
Sir, mRNA translation.
Translation from mRNA. So how mRNA is synthesized? Transcription. So transcription occurs from what? DNA. So in DNA there are some things which are converted to proteins. So what are those things? Genes. So how many in a normal human cell, how many genes are present? How many genes are present? Around 25 or 2000. So when we want to identify the structure of a protein or a polypeptide, we can also do it from the gene or from the mRNA. If we can identify the structure of mRNA, we can identify what will be the amino acids that can be inserted one after another. So for that, what we have to know? We have to know this genetic sequence or the structure of DNA or mRNA. And when we know that, we can identify the structure of a protein. But which one is preferred? The previous method of identification of a structure of a polypeptide, or this one, which we can identify from mRNA or from the genetic sequence of a DNA. So which one is better? First, we can identify the structure of a protein by dissolving all the peptide bonds, then estimating the amino acids, and then we can also find the sequence in the lab from a protein. And another way is we can identify the structure of a protein from the sequence of a gene. So which one is better? Any guesses? Someone said gene. So if it is gene, why? Like, what are the advantages? There must be some advantages. And if it is from polypeptide, there must be some advantages.
You don't have to dissolve the peptide bonds.
But you have to determine the sequence of the genes in the DNA. So we generally prefer by dissolving the peptide bonds, because there are several steps in the formation of a protein, like transcription. There are also translation. And when mRNA is synthesized, it also has to pass through several steps. And there are also modifications after a protein is being formed. The amino acids can also get changed. So this change we cannot determine from the genetic sequence, like what will be the ultimate protein and if there will be any change or not. But when we are directly determining the sequence and the number of amino acids from the polypeptide chain, that will be more accurate. Understood? So here we are analyzing the lab. Now there can be many big proteins. So there can be also some proteins which can have several polypeptide chains. So a protein, when it is having several polypeptide chains, what kind of structure it is? I told you about four structure: primary, secondary, tertiary, and quaternary. So when a protein is having more than one polypeptide chain, it will be quaternary structure. So when we are determining the structure of a quaternary protein, it is sometimes difficult because the protein is bigger in size. So what we do? We will separate the different polypeptide chains, and even sometimes the polypeptide chains are really big in size. When we consider a polypeptide chain more than 100 amino acids, so then it is sometimes difficult. So we sometimes cleave the polypeptide chain with enzyme. The enzyme can cut a polypeptide chain at specific sequence or at specific location. So suppose a polypeptide chain has 150 amino acids and we are using one enzyme. Suppose enzyme A. And we know that this enzyme cuts the polypeptide chain when it sees the amino acid alanine or glutamic acid. We know that. So we know this enzyme will cut this polypeptide chain at that part only where it sees a glutamic acid or valine. Suppose we are using this and cutting this polypeptide chain of 150 amino acid into two parts. And we are using another enzyme, enzyme B. And this enzyme B have also a specific sequence or location where it will divide the polypeptide chain. Suppose it cuts the polypeptide chain into 3 parts. So then we will take the 3 parts and sequence it. The 3 parts have been done by the enzyme B. And the same polypeptide chain has been divided into 2 parts by enzyme A. So now we are analyzing these 2 parts and these 3 parts separately. Then it's like jigsaw puzzle. When all the data are available with us, then we have to sit down, do permutation combination, what are the segments that can be attached. And then we will find out the actual structure of the polypeptide chain. So here we can see we are using the enzyme TRPC. And what this enzyme does, it is cutting the polypeptide chain at 2 places, where it says lysine and arginine. And then here we have, from one big polypeptide chain, we have 3 small polypeptide chains. So here we can do all the permutation combination and see what can be the arrangement. And then we are using the same polypeptide chain and using some other chemical agent or enzyme. Here we are using cyanogen bromide. And where it cuts, there must be methionine. So this, the same polypeptide is cut at one place, and it is divided into 2 parts, peptide X and peptide Y. And then we just solve by this and this. If the polypeptide chain is much longer, we can use more enzymes. And with the help of this data from the computer can itself arrange and tell us the sequence of the actual polypeptide chain. So most of these things are generally used with auto-analysis. And this thing we have already discussed, how we can determine the structure of a polypeptide chain from a DNA. So this is the DNA, this is RNA, and this is protein. So can anyone tell me this sequence, what it is called? Central dogma. So this is less favored because the post-translational modifications we cannot determine when we are determining the structure of a protein from the genetic sequence. Okay, from next class we will continue with detailed structure of the secondary structure. First let me tell you the overall idea. So we generally have to read the 2 secondary structures. One is alpha helix and beta sheets. So they are just when we are building a large building, we have to make first rooms. Before that, we have to make the walls. So when we are building the walls of a room, what we use? We use the bricks. The bricks can be compared with the amino acids. So during building the wall, we first make holes. So when I am seeing you guys, so in this class, if I am here standing in the front, so which are the rows and which are the columns? Yes. So in this class, if I am standing here, these are the columns. So there are 4 columns. and these are the rows. So in the wall, we have to first make the rows to build the height. So what we do, we just place brick one side by side to make the rows. So this side by side arrangement of the bricks, and when we are connecting them with the cement. So this is the primary structure. So once the wall is formed, so there must be connection between one row of bricks with the row above and below the row of bricks. So this structural relation with the different bonds, this is the secondary structure. And after the wall is formed, suppose this whole wall has been formed. When there is connection between this wall and this wall, there must be something connecting. So this connection in the structure of protein can be done by loops, bends, turns. So when these different secondary structures are connected with bends, loops, turns, they are called super secondary structure. So after this super secondary structure has been formed, that is a room with the four walls. To stay in this room, we need the ceiling to be done. When the ceiling is done, it is a complete room where we can stay. That is the functional room. When this functional room is formed, it is called tertiary structure. That is the functional structure of a protein. And when one room, another room, several rooms are formed, we are forming a building. When the whole building is formed, that is the quaternary structure. So next class we will start with the detail of the secondary structure.